So, welcome back to the 6th week of this course. So, we are into the second lecture of the 6th week. So, in the last lecture, I talked to you about one of the very prominent assays where we showed that how LFA, lateral flow immunoassays work. So, when we talk about immunoassays in general, the idea or the logic is pretty straightforward. So, you have say a toxin, and against that toxin you raised antibodies or any kind of marker. Okay. And what you do is you put the marker or immobilize the marker. Okay. This immobilization step is of extreme importance, and on the immobilized plate you roll the toxin. Okay. So, when the toxin binds to the substrate, there are multiple ways you can read. So, say for example, once due to the binding there is a change in its conformation, you measure the conformational changes, or what you do, there are some kind of fluorescent marker which gets activated upon binding of the toxin. Yet there are other ways where you try to see a competitive binding, and that is where we talked about the competitive binding last time, where you have say very similar to toxin another antigen which is weak binder. Okay. It binds, say for example, this is the weak binder, it goes there and weakly bind on the surface. So, now when you put the toxin, it competitively displaces the weak binder and thereby leading to what we call as a competitive assay. So, that is where we completed the last class about the competitive assay, and where we talked about competitive assays. There are two main types of competitive formats. In the first type, the target in the sample and the labeled target, alternatively a molecule with less affinity for bioreceptor than the target itself, in the conjugate pad compete for test line capture bioreceptor. In the second type, the target in the sample and the target on the test line, which would act as a test line capture bioreceptor, compete for the labeled bioreceptors. Okay. So, this is the competitive assay what we talked about. From the competitive assay, today's class will be sandwich or non-competitive assay. This is the topic for today's class. Okay. A sandwich or non-competitive assay is probably the most used one. This is the most popular one strategy for the detection of mid and big size analytes. So, remember when we talked about the competitive assay, we talked about things which are less than 1 kilodalton. Okay. But here we are talking about more than 1 kilodalton, which is typically most of the molecules are at that zone of more than 1 kilodalton, okay, such as proteins, antibodies, bacteria and cells. So, one thing is very clear, the sandwich assay is for bigger entities, which includes the proteins, whole protein, antibodies, bacteria and the cells in LFAs. If it function by capturing the large molecule between the detection bioreceptor and the test line capture bioreceptor, produces a signal that increases proportionately with the amount of target in the sample. So, it is kind of the density of the signal or strength of the signal will increase as more and more of these molecules are getting captured. Okay. So, having said this, I will take you back a little bit into the general scheme of things where you will get an idea. So, when you talk about the signal output, so when we are doing this kind of assays, actual signal output will be either a fluorescent probe or a change in color or some kind of electrochemical signature or something. Okay. So, there has to be some signal output. So, whenever I am talking about there will be a change in the signal output, you have to realize something has been conjugated to that particular site, which either glows or it quenches, it glow quenches. Either way, there could be an increase in the fluorescent signature or could be the decrease in the fluorescent signature. Similarly, there will be increase in color formation or decrease in color formation. Similarly, there will be more electrochemical signature or less electrochemical signature. So, whatever way, there has to be a significant change which is supposed to be observed before you can make a claim. So, whenever we talk about, so I told you, so this is the target reaches here, there is a binding, this binding is being given by a signal output. So, this is how it works. That is why there are 4 segments: target, recognition element. So, in this case, the recognition element is say antibody which is embedded on this. So, what I was trying to tell you guys was that antibody is being immobilized, and on these immobilized surface you run your target molecule, and upon binding what you see is a signal output. These are the 4 basic element of a biosensor configuration. Okay. So, now coming back where we were. So, from here we are moving on to the bigger entities, which is a non-competitive assay. So, in sandwich or non-competitive assays, one key aspect for the development of this type of assay is the requirement of 2 bioreceptors that bind a different portion of the target. So, what exactly that means? That means, let us take a simple analogy. Whenever you go to a doctor and doctor says that, say you are suffering from, say, diarrhea, or you are suffering from some disease, what we do, your friends will tell you that take a second opinion, talk to another doctor, let us see what that doctor says. So, in a way, in order to confirm that you really suffer from that disease X, you are taking multiple opinions. Exactly the same thing. A molecule has multiple binding sites. So, say for example, molecule A, talk about molecule A, and suppose this molecule is something like this. So, it has a binding site out here and it has a binding site out here. Okay. There are 2 binding sites, binding site 1, binding site 2. Say for example, another molecule which is B, which is very similar in structure, except it has a very similar site as out here. So, this site what you see out here and this side what you see are similar, but now you want to detect exclusively A molecule, not B. So, what will happen if you only rely on this particular binding site? Then there is a possibility that there may be a false reading. So, in that case what you do, you use 2 recognition site, that is what it says, 2 different monoclonal antibodies or a labeled monoclonal antibody and a captured polyclonal antibody. So, you need 2 different spots where your configuration, if you go by the configuration, it will be something like this. For this, say for example, I am talking about this vegetative cell, I need it to bind to say 2 different kind of fragments of monoclonal antibody, say MAB1, MAB2. So, if MAB2 is represented by like this and MAB is represented by like this. So, what you will see in the cell will be, there will be site, one second, there will be site where it will be binding like on this, and there will be site where it will be binding like this. So, you need 2 spots for it to bind. So, LFIA plate will have both immobilized MAB1, MAB2. You can have even more than that if you want, in order to have a complete confirmation that this is your target, because you cannot afford to have a false reporting of the target. You have to be very clear that this is your target, and that is why they use 2. It functions by capturing the large molecule between the detection bioreceptor and the test line capture bioreceptor, producing a signal that increases proportionally with the amount of target in the sample. So, one of the major requirement is 2 binding sites. So, in other words, we are talking about 2 antibodies which bind to your target. Okay. For example, 2 different monoclonal antibodies or a labeled monoclonal antibody and a captured polyclonal antibody are the most secure way to ensure the sandwich formation. in this case is particularly a big target. So, in a way what you are doing is that if this is your one monoclonal antibody and this is your other monoclonal antibody and here is your target. So, you are sandwiching the target between monoclonal antibody 1 and monoclonal antibody 2. So, this is where your sandwiching is happening, and most of these big molecules indeed get sandwiched between these two targets, such as bacteria or cell, where the same antigen is repeated many times in the same target and the use of the same antibody, better polyclonal, would be also feasible options. Okay. Now, talking about a little bit more about the sandwich assays. Sandwich assays are subjected to possible hook effects in case of extremely high target concentration. So, I will request you guys read about what is the Hooke's effect. Otherwise, in future classes, I will again explain this part. Okay. And they are more prone to false positive results than competitive assays. For their optimization, high concentration of test line capture by receptors are generally recommended. So, that means you have to go for 1 milligram per ml. That means when you are talking about this kind of concentration, that means your need for the antibodies will be far more higher versus when you are doing a competitive binding assay. For non-competitive sandwich binding, your antibody requirements will be far higher, okay, to maximize the chances of a sandwich formation, because you need more dense concentration. Now, if you look at the schematics out here, something like this, here you have the sample pad, you have the conjugate pad, you have the test line, and here you have the control line, you have the adsorbent pad for the other side. Okay. So, this is where the binding is happening. Look at it. Schematic of super paramagnetic lateral flow immunoassay. So, this is where the retention line is happening, monoclonal antibody raised against Bacillus anthracis. So, this is being used for Bacillus anthracis spores, and this is how you generate the signal using some kind of magnetic reader, which is, that is why it is called the super paramagnetic lateral assay, because the binding leads to change in paramagnetic signal. So, as I said, there are multiple ways you can read it. There could be electrical signature, there could be magnetic signature, which we generally do using EPR or ESR, electron spin resonance, or electron EPR signals, or we do it with fluorescence assays, we do color assays, but these are all different reading techniques, how you can read that. Apart from it, we have talked about other techniques in the previous classes, but end of the day, the basic understanding is it's a simple immunoassay technique which is being further modified using on a microfluidic platform. And mind it, these are not newer techniques, it is just the miniaturization is the newer part. Otherwise, radioimmunoassay, RIA, or ELISA, enzyme linked immunoabsorbent assay, these are all old techniques. These are there for almost now 60 years, but the real improvement or the real, I will say, change which has happened in this area is by virtue of miniaturization, antibody production. So, remember, first is miniaturization, use of MEMS technology, micro electromechanical systems, use of microfluidics, use of advanced optics. Then there is huge amount of precision detection in terms of electrical signature, magnetic signature, fluorescence signature, color signature, and which has been assisted by advanced optics technology. And on top of that, the hybridoma techniques, and other techniques of raising antibodies. So, in all, if you look at the whole thing, it is an integrated approach of developing devices which are handheld, miniaturized. So, some of the basic feature what is needed is they should be transported, they should be handheld, easily packageable, they should give precise signature, and they should be things which could be mass produced. Those of you who have seen in television or other about pregnancy test, these are small kits, it's exactly like that. So, we have seen a lot of such development of these kind of assays during COVID, where you are detecting. So, all these things are now happening at the level of technology. Much of it is to do with micro manufacturing. So, this is the other aspect which is of extreme importance for developing these kind of sensory system. But you realized one thing, and there is one more thing which I forgot to mention, is conjugate chemistry. This conjugation chemistry is of important because when you have to conjugate the antibody with fluorescent probes, you have to conjugate the antibody with some enzyme for enzyme link immunoabsorbent assays, or you have to conjugate the antibody with some magnetic probe, or you have to conjugate the antibody with some electrical probe, or you have to conjugate the antibody with some other dye. So, there is huge amount of conjugation chemistry which is happening. So, as I was mentioning in the previous lecture, the whole field of designing a biosensor is a very integrated approach. So, you have to first of all understand the basic biology and chemistry of binding assays. So, that means if you have to detect a toxin or a spore or a pathogen, you have to know to what it binds. Either you raise antibodies against it, or you have some synthetic target to which it will bind. Then you have to create an assay platform first of all to see what is the level of detection, how low you can go, what is the minimum amount of titer you needed. When you talk about the minimum amount of titer, we mean that what is the lowest concentration you can detect. So, this takes me to some of the very early studies where there are a lot of controversies which erupted. Back in the year 2000, I still remember that in NIH there was a huge amount of National Institute of Health in the United States. There was huge uproar that cancer detection could be done on a chip by virtue of the fact that any protein or any biomolecule which is getting generated in our body due to the presence of a cancer could be detected with even a small pint of blood or a small drop of blood. There was a huge amount of investment which happened. As a matter of fact, one of the biggest controversies of that time erupted to develop such detection devices. But the problem what was simmering is that you cannot detect extremely low amount of proteins without lot of noise, and this was a huge problem. You can go down, but it will come with a set of huge amount of noise. And knowing the fact, all the protein assays which were there say 25 years back, none of them, and even now also most of the protein assays cannot go because you will see a lot of noise. With that promise, lot of investments were done, and these investment did help the field to grow definitely, but still now detection of extremely low concentration of protein is a challenge in the real life. You could have published paper here and there. I have seen many of those, but the fact is when it comes to scaling up such technologies, that's where it fails. So, this is one of the challenges you have to realize, that when we talk about smaller number of molecular detection, every detection element comes with its set of noises. So, you have to realize that any of these what we are teaching today will become old in next couple of years or next 5 years. So, we need newer and newer ways to detection without noise, and that is the challenge. Whatever I am writing, this is people have been working on all these for last many decades, but still the challenge comes how low we can go, what will be our lowest detection limit. So, keep this fact in mind. There is enormous room of research or to develop career in these areas, provided you understand the fundamental basics and you understand how you can cut down on noises with more and more innovative techniques. So, I will close in here. In the next class, we will move on to the botulinum toxin. Thank you.